Hi, this is Rethik, your biology subject matter expert from MEMONEET. And once again, welcome to the MEMONEET series. So in this unit, we are actually going to discuss on the major chapter that is the unit of life, which is the cell. So what do you mean by cell? And when coming to cell, right? So when we look around, we see both living and non-living things. That means both the living things as well as non-living. So one thing is very much sure that all the organisms, they are composed of cells, and some of them are composed of single cells. Okay. And some of them are composed of many cells. Say like if it is single cell, and some of them are actually composed of many cells. So multicellular, like us, we are multicellular organisms. Then single cells such as bacteria or individual cells and all. Then what is this cell? Since we get to know that all the living organisms they are composed of cells, then what exactly is the cell? See, cell, they are independent existence performing an essential function of life. Anything which is less than a complete structure of a cell does not ensure independent living. That means if a complete structure is not made up of a cell, then obviously that living cannot be said as they're independent. That means all the cells, they perform their individual function and they group together to form a specific function. Not only that, all the living organisms, they are made up of different kind of cells. Hence, with that we can define as cell as the fundamental structure and functional unit of all the living organisms. So very first, the living cell was described by Anthony van Leeuwenhoek. Later, the nucleus in the cell was discovered by Robert Brown, and with the invention of this microscope and also the improvement that led in the electronic microscope has revealed the structure detail of the cell. Based on the structure, based on their function, the cell theory has been proposed. In 1838, Matthes, a German biologist, he examined a large number of plants and also observed that all the plants are composed of different kind of cells, okay, which forms the tissues of a plant. Like if it is a plant leaf, he got to know that they are composed of what? Different kinds of cells. Say like in leaves, there were different kinds of cells. In stem, there was different kinds of cells all together, okay, which forms the tissues of the plants. So that is the cell is what giving the structure. That is what he got to know. Next, later on, in 1839, Stephen, one of the British zoologists, he studied different types of animal cells here. So the back, they studied what? The plant cells. Here, they studied different types of animal cell. And later they reported that the cell has thin outer layer. Okay. So if this is a cell, so there is a thin outer layer which is surrounding around the cell, and he named them as what? Plasma membrane. So later on, based on that, he proposed an hypothesis that the body of animals and plants are composed of what? All the body of animals and plants, they are composed of one thing is cell and the another one is product of what? Cell. So that is what he got to know. Clear? Next. In 1855, Virchow he did a very first explanation that the cell divide he gave a very good explanation that the cell, if this is an individual cell, so that cell gets divided and a new cell has been formed from it. Okay, a new cell has been formed from what I can say pre-existing cells, and he named it as omnicellulae e cellulae. Okay, that means so before there was a cell. So from that cell, a new cell was been emerged. So that it means. Then he modified the hypothesis of Sheldon and Stephen to give a cell theory a final shape, where he gave a proper cell theory. So before the one, this scientist who studied on plant cell and he studied on animal cell, based on their hypothesis, he merged, he gave a proper clarity, and finally he proposed a cell theory in a fine shape. So cell theory was actually understood today is like the what exactly the cell theory we follow today is all living cells are composed of cells and product of the cells. Okay, this is one point. That means all the living organisms are made up of cells and the product of cells. Then all cells actually arises from what? Pre-existing pre-existing cells. That is what he meant to be. Clear? Now, so if you take an onion peel, you do have an experiment, I guess. So if you take an onion peel and if you absorb the cell, okay, a typical cell. So what you will be absorbing? You are going to absorb a kind of rectangular shape here. Okay. So that means the onion cell, which is typically a plant cell, has a distinct cell wall, its outer boundary. So here in the outer boundary, you are going to see a distinct cell wall. Okay. And just within it is a cell membrane. So here within this, what you're going to see, cell membrane you're going to absorb. The cell of the human chick, that is the egg. Okay. So have another outer membrane as the deliming structure of the cell. So inside each cell, there will always be a dense membrane bounded structure, which is actually called as nucleus. And this nucleus, very important thing is, it contains what? It contains the chromosome. So this chromosomes, it has what? The genetic material, which is actually called as the DNA. So here, students, DNA is the genetic material. So cells that have membrane bounded nucleus, that means if they have a membrane bounded with the nucleus, we normally call them as eukaryotes, where cells that actually lack this membrane bounded nucleus, that means if it is a nucleus, it should have a membrane. So if it does have a membrane, we normally call it as eukaryotes. And those which has no membrane bounded nucleus, they are actually called as what? Prokaryotes. Clear? We'll discuss one by one what is prokaryotes and eukaryotes in detail. But for right now, you can see this is an anatomy of animal cell and this is the anatomy of plant cell. So in animal cell, you can see a very well-defined structure of the nucleus, membrane bounded. Okay. So whereas in plant cells, you don't see such well-defined structure of the nucleus. So membrane bounded nucleus, you cannot see. So that is the major difference. So in growth prokaryote and eukaryotic cells, so you can see there is a fluid which has been filled here. So we normally call them as what? Yeah, cytoplasm. So what happens is all the fluids. So this is cell membrane. You got to know the outermost layer. So the fluid which is filled, right? So we normally call them as what? Cytoplasm. Okay. So the blue color and the yellow color in plant that you're watching is actually a cytoplasm, and it normally occupies a very volume of the cell. That is the large volume of the cell. The cytoplasm is the main area of the cellular activity. That means inside the cytoplasm, the cellular activities will happen. Okay. So in both the plant as well as in animals, inside the cytoplasm itself, the cellular activities will occur. Various chemical reactions and all will happen inside the cytoplasm itself. So this cytoplasm is responsible to keep the cell in the living state. Okay. So beside the nucleus, eukaryotic cells have other membrane bounded distinct structure. So I can call them as organelles. Like, have you seen endoplasmic reticulum? So we have here vacuoles, lysosomes, mitochondria. Okay. And the nucleus is there. Then we have endoplasmic reticulum, Golgi complex. All these things are there. So here it has Golgi complex, lysosomes, mitochondria, microbodies, vacuoles, and all. Okay. So the prokaryotic cells, they lack such membrane bounded organelles. I'll explain you how. So one thing you're very much clear with one thing is in plant and animal cells, we have a specific nucleus will be present. Okay. But I'm just saying you are denoting like how different the nucleus is. Okay, in plant cell and animal cell, you will definitely going to observe the nucleus, nuclear structure, nuclear membrane, everything, but the structure is different. Okay, but that all comes in eukaryotic cells. So here, plant and animal cells are obviously eukaryotic cells. But in prokaryotes like bacteria and all, you won't see such membrane bounds, nucleus structure and all. So if this is an animal cell where you are observing what? All these things. But in bacteria, you will see no such boundage of the membranes. So we will go one by one, like what all those, not in detail, because I'll be explaining in detail in the coming video. So first thing that you will observe is there are another organelles, which is like ribosomes. So now ribosomes, they are non-membrane bounded organelles, which actually you will find in both the prokaryotes as well as in the eukaryotes. Then within the cell, what happens? The ribosomes are found not only in the cytoplasm, but also within the two organelle chloroplasts, that is in plants, and mitochondria or rough endoplasmic reticulum. So they are not only you will see them in the cytoplasm, but also between the two organelles, also within the sorry. That means within the two organelles. Like if it is in plants, we can observe that in the chloroplast. And if it is in animals, we do observe these ribosomes in mitochondria and in rough endoplasmic reticulum. What is the function of ribosomes and all? We will discuss it in depth. Okay. The next is we have the centrioles, even that is non-bounded, like animal cells, contains another non-membrane bounded organelles. So that is actually what? Centriole, which actually helps in cell division. Then cells differ greatly in their size, shape, and also their activities. For example, mycoplasm is a cell which is actually a smallest cell. This is mycoplasm. It is very smallest cells which measures about 0.3 meter in length. Okay. While in the bacteria is another cell which is again measures about 3 to 5 meter in length. So the highest cell is actually the ostrich cell. Even it is considered as cell, it is the most largest and the big cell. Okay. So cell, I told you the structure is also different. Say like we have red blood cells, which is round and concave in structure, white blood cell, which is amoeboidal in structure, and columnar epithelium, long and narrow in structure. So where this RBC cells, they are human red blood cell, they are about 7.0 micrometer in the diameter, and we do have this nerve cells, so which is what, branched and long, right, which is the longest cell that we say. Then in human, so we have this also tracheal cells, which is elongated cylindrical. Mesophile cells are present, which is round and oval in structure. So before coming to eukaryotes, which is well established, well structured and all, first we will see about the prokaryotes. So today's video we will be discussing till prokaryotes. Then I'll be discussing completely about the eukaryotes. Okay. So in prokaryote cells or prokaryotic cells are the cells that do not have a true nucleus and membrane bound organelles. So this is first point. So the prokaryotic cells are they are been represented by what? Bacteria, blue-green algae, mycoplasm, and pneumonia-like organisms. That is PPLO. So you can see here, which is prokaryotic cells here. So they are generally very small, then multiply more rapidly. Bacteria multiplies more rapidly when compared to the eukaryotic cells. So they may vary greatly in the shape. Like one prokaryote, one bacteria might vary from the other bacteria or green algae. So the cells shape they vary. Right. So, but the four basic shape of the bacteria are the rod-like structure, which is actually called as bacillus, coccus, which is spherical-like structure, then vibrio, which is comma-shaped structure, and the spirillum, which is spiral-shaped structure. If you remember the biological classification, you'll understand it. Then the prokaryotes, they have a cell wall surrounded by the cell membrane, except in the mycoplasm. Then even mycoplasm is an organism which doesn't have the cell membrane. Then the fluid matrix which is filled inside the cell is actually called as what? So it is called as the cytoplasm. Then you cannot see any defined nucleus, right? So that means well-defined nucleus you won't see. The genetic material is basically naked genetic material. DNA, it is like this, obviously. Naked genetic material, not enveloped by a cell membrane. That is, it is not covered, surrounded by a cell membrane. In addition to the genetic genomes, that is the DNA, so there is, like I told you, if this is a bacteria, right? So additional to the DNA, so there is another one thing which is actually called as circular DNA or single chromosome. So many bacteria have small circular DNA outside the genome DNA. So these small circular DNA is actually called as plasmids. So the plasmid DNA confirms certain unique phenotypic character to such bacteria. So one such characterization of the plasmid is it shows what? Resistance to the antibiotics. So there are antibodies, right, in our body or few in even in environment. So it shows resistant to such things. Okay. Next up. So nuclear membrane is found in the eukaryotes. No organelles like the ones in eukaryotes are found in the prokaryotes. That is nuclear membrane and any other organelles, right, which are found in the eukaryotes. You don't find them in the prokaryotes, but except ribosomes, you will find in both prokaryotes and eukaryotes, as I told before itself. Then prokaryotes, they have something unique in the form of uh, very unique, okay, inclusions, such as a specialized differentiated from of the cell membrane. They are called as there is presence of mesosomes, which is characterization of the prokaryotes. They are essential foldings of the cell membrane. Next, So one thing we will try to understand or figure out is the envelope and its modification, how they got modified, as I told you. So most prokaryotic cells, particularly the bacterial cells, okay, so they have a chemically complex cell envelope. The cell envelope consists of tightly bounded three-layer structure. So one is outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Okay. So if this is the bacteria cells, you will actually see a bacteria cells. The outermost cell envelope consists of three layers. Glycocalyx will be the outermost, then by the middle, you will actually see the cell wall, then the plasma membrane. Then, although each layer of the envelope perform different function, they act together in a single protective unit. So, all the layer they do perform a different function, but together, they act as a single protective unit. So, they can be classified like bacteria can be classified into two groups based on the presence or absence of envelope. Okay, based on the differences in the cell envelope again, like a manner in which they respond to certain strain produced during the gram positive or gram negative, something like that. Okay. So those that take up the gram strain are actually called as gram positive bacteria, and the other which do not take the gram strain are actually called as gram negative bacteria. So the outermost layer of the bacteria, which is glycocalyx, differs in composition and thickness among different bacteria. So it could be loose sheath. They're called a simple, slim layer. A slim is nothing but a kind of mild layer, which is other, it may be thick, like from organism to organism. Say like from one bacteria to other bacteria, the outermost layer that we are talking, right? So that might get differ, like the calyx. It differs in its composition and thickness. Okay. So another thing is, while in other it may be very thick, in some it may be tough, and in some it may be thin. So we normally call them as what? Capsule. So the cell wall determines the shape of the cell and provides a strong structure supports to prevent the bacteria from bursting out or by collapsing. Then we have the plasma membrane, so which is semi-permeable in nature and interacts with the outside world. So this membrane is simple structurally to that of the when compared to the eukaryotic cells. So the plasma membrane in prokaryotic cell is very simple. Then there is a special membrane structure, which is the mesosome, which is formed by the extension of the plasma membrane into the cell. So these extensions are in the form of vesicles, tubules, and lamellae. So what are the major function of these things? So they help in the cell wall formation, one point, then DNA replication, then distribution to the daughter cell. Like when the bacteria, it divides, right? So it has to distribute this DNA equally to both the daughter cell. So that process is also being carried out. So they also help in respiration, then secretion process to increase the surface area of the plasma membrane, and also many enzymatic content will also be present. So in some prokaryotes like cyanobacteria and all, so there are other membranous extensions into the cytoplasm. So they are called as chromatophores. Okay, they are called as what? Chromatophores, which contains a certain pigmentations. Right. So the bacteria cells may be motile or non-motile, depends whether it has flagella or not. So if they have a thin flagella, say if it is a bacteria, right? So if it has this thin flagella extension from the cell wall, so that we filamentous. So we normally call it as flagella. So bacteria, some of them shows only one flagella, some shows two flagella, and some has arrangements across its body flagella. So the major function of this flagella is the momentum. Okay. So flagellum is composed of three parts. One is filament, hook, and basal body. So the flagella is the longest portion and extends from the cell surface. As you can see, it has been extended from the cell surface to the outside of the body. Beside the flagella, we also have the pili and fimbriae. Okay. So what is the major function of pili and fimbriae? So they are also a surface structure of the bacteria, but they don't play in the role of called mortality, that is momentum, but could help in the exchange of the genetic material from one bacteria to the other bacteria. Yeah. So here the pili are elongated tube structure made up of special protein. The fimbriae are small, breast-like fibers which is surrounded or sprouting out of the cell. In some bacteria, they are known to help in attaching the bacteria to the rock in streams and also in the host tissue. That is the bristles that I have been talking. Okay. So this was all about the prokaryotic basic things, and it's very simple since you won't see such organelles, complicated organelles in prokaryotic. So in the coming video, we will also discuss what is eukaryotic cells and different organelles which are, comes under this eukaryotic cells with proper functions and all. So for now, if you do feel any doubts, you can reach me out through the comment section or else you can join our Telegram channel and I'll be also available there. Thank you.